Okay, sir. So we are live now. So let us. So let me. So we will start today's proceedings. So once again, uh, myself, Dr. Naveen Gupta, I welcome all of you in the Sunday lecture series. Uh, as I have explained earlier also, we are doing this lecture series with multiple purposes. We are in some of the lectures, we are calling some very senior peoples so that we can benefit from their ideas and from their experience. In some of the lectures, we are giving opportunity to the young faculty and the students also so that they can share their new ideas and they can also get used to this online lectures and all that. Now, in this regard, today we are having a very young researcher, Dr. Sonia with us. And we are very lucky that she is also a alumni of our department. And we have her teacher, Professor Sanjay Chibgar, the senior most professor in the Department of Microbiology, is also here. So we thought that we will, in most of the lectures, the students will be there. We will call the teacher so that they can share their memories, so that it is a little motivating for the students. So with this background, I call Professor Prince Sharma to welcome the all and start the today's lecture. Professor Prince Sharma, please.
Um, Thank you, Naveen. You have said it very rightly that Association of Microbiologists of India, Chandigarh unit, is organizing these Sunday lecture series, and in this series we are calling people who are very senior, very eminent scientists, but on the same uh, side we are calling very young researchers also, young faculty and research scholars also to present their work because we feel that some of them who are doing very good work and who are budding scientists, they should also be given the platforms to expose their work, and they can be brought to limelight, and they can be also heard uh, about what kind of work they are doing. So it is always the prerogative of AMI, Chandigarh unit, that we are to promote microbiology, we are to spread microbiology, we have to bring microbiology to the general public, and as far as touch all those pertinent issues which are of importance to the society. And in this line, now today's lecture will be something very interesting on the biofilms. And later on, she will be introduced. So Dr. Sonia is our own alumnus, and she's working as a microbiologist in a dental college. So it will be interesting to hear her research work. So I welcome her to our family of the Association of Microbiologists of India in this Chandigarh unit, and I welcome her mentor, Dr. Chibar, and he will be now introducing her formally to us, and he will be telling more about her to us, all of us, and he will be also taking part in the discussions following her lecture. So welcome, Dr. Chibar, and I also welcome Dr. Kalia here. He who is uh, a, a great scientist, former IGIB director, and our great friend. And he is from South Korea, and we are happy that he is joining us in this webinar series. And he will be also putting in some of his valuable suggestions and guidance to the young researchers like Dr. Sonia. So I welcome him, and I welcome all my audience here who are here on this leisurely Sunday. They are here to listen to this lecture. So welcome you all again. A very good afternoon, and I hand over now this to Dr. Naveen to carry on the proceedings.
Thank you, sir. So, as I said, today we have Professor Sanjay Chibbar with us. He is a teacher of most of us who are present on the screen, what you are seeing. So he has spent whole of his life doing microbiology in our department. I am very lucky to we are very lucky to have you here, sir. So I just now
Thank you.
request Chipper sir to share his memories with Dr. Sonia as a student because he was directly related to her, and also introduce Dr. Sonia to all the people who have joined us. Chipper sir, please.
Thank you, Dr. Naveen. First of all, I congratulate the Chandigarh chapter of AMI because you are doing a very good job, and especially you are providing a platform to the young scientists also, because generally they don't get an opportunity to showcase their work and discuss their things, and this is something very good opportunity you are giving to the young scientists. I'm very happy on this part, and I congratulate you and your team who are doing this good job. Um, Coming to today's speaker, yes, it is. I'm very happy to introduce Dr. Sonia Bhonchal. She, as you already said, she is an alumnus of our department. She did her graduation in 1995, then post graduation with thesis under my supervision in 1997, and then she moved to PGI Chandigarh to do her PhD, and she completed her PhD in 2007, and after that she got selected in the Dental College of Panjab University Chandigarh, where she is working at the moment as senior assistant professor, and she has published in good journals around 28 publications, book chapters. She is also having projects funded by the funding agencies. So she is doing very well, and she is a very good teacher, I know, because she is quite popular among her students. She is teaching the BDS and the MDA students and helping them also in their research. That is something good as far as Sonia is concerned. And she has started working on biofilms, and her interest in biofilms is because biofilms, they play an important role in the dental infections as well. And I think this is the opportunity where she will be able to tell us about her interest today, and I wish her all good luck. And I think Sonia will take over and deliver the lecture today. Thank you.
Should I start?
Dr. Naveen?
Thank you.
Yes.
Thank you, sir.
Huh.
Good morning. Yeah. Good morning, chairpersons, Professor Chibar, Professor Kalia, and Professor Prince Sharma, Professor S.K. Soni, Dr. Naveen Gupta, and all the faculty and students. And as I'm being introduced already, that being the alumni of this department today, I'm feeling really blessed and happy to get this opportunity. And Professor Chipper, as he has already introduced me, that he was my PG guide, and I'm getting his guidance till date, and his constant encouragement is really motivating for me. And he is really scientific par excellence. I'm also thankful to the organizers, Professor Prince Sharma, Professor S.K. Soni, and Dr. Kusum Harjai, a very dynamic coordinator, Dr. Naveen Gupta, for giving me the opportunity to share my presentation and in this lecture series of Micro Department of Microbiology, Panjab University. So I would like to start with my presentation now.
You can share the screen.
Right, sir. Is it clear, sir? Is it okay?
Yeah, it's okay. It's okay.
Fine, sir.
So,
Yeah.
yes.
Right, sir. So my topic for today's presentation is microbial biofilms, development, pathogenicity, and role in oral health. Um, I've chosen in this topic as I'm quite interested in this field, and in this presentation, I'll be elaborating more on dental plaque as a biofilm and the oral diseases it causes. And what I wanted to highlight in today's presentation is though I am working in the dental institute, but what are the new researches which are coming up, which are linked with biofilms, oral biofilms, and the disease they are causing, and how we can prevent them. As you have been already introduced, like I'm working in Dr. Harvansh Singh Judge Institute of Dental Sciences and Hospital, which is almost a block away from the Department of Microbiology, Panjab University, for the last 13 years. And more or less since these 13 years, I have developed an interest in oral microbiology. Okay, I get my laser pointer now. Uh, Let me start now. Yeah. What is a biofilm? Some of the things are targeted at a basic level. A biofilm is an aggregate of microorganisms in which cells are stuck to each other or to a surface. These adherent cells are frequently embedded within a self-produced matrix of extracellular polymeric substance, or EPS. Now, this can be acquired from the environment or the bacteria themselves. Biofilms exist everywhere. If you see, they exist on medical devices, medical implants on patients, in the environment, in rocks, in rivers, streams, and in mouth. And these organisms in the biofilm, they are very large number and a different organisms, which makes it a very challenging field to study. Biofilm supports the microbial growth, as biofilm is a common mode of bacterial growth in nature, such as sewage treatment, corrosion of materials, food contamination, pipe collapse, plant microorganisms interaction in the biosphere, the formation of dental plaque, the development of chronic infections in live tissue like mastitis, otitis, pneumonia, urinary infections, osteomyelitis, or problems related to medical implants are also created by biofilms. Now, I'll be discussing more of biofilms as a cause of nosocomial infections and microbial biofilms. One of the main property which makes the biofilm study a challenge is that they are formed by antimicrobial resistant organisms. And almost 65% of infections treated in the developed world are the ones which are caused by these biofilms, microbial biofilms. Biofilms acquire the property of antibiotic resistance, as the EPS which the bacteria secretes is believed to play a role in some antibiotic resistant infection. Biofilms, and this also makes them, these biofilms need a higher concentration of antibiotics. The bacteria within the biofilms have increased up to 1,000-fold higher resistance to antimicrobial compounds. Even though if you see that the same bacteria are sensitive to these agents if grown under planktonic conditions. Now, another thing about biofilms is the antimicrobial agents which are administered during valve replacement and whenever the patient has dental work to prevent initial attachment by killing all microorganisms introduced into bloodstream. Because when any patient is, for example, is having dental work, these biofilm may get, some of the cells may get detached from the site and they may enter the bloodstream. So it's very important that these organisms are killed. Biofilms help gene transfer. And the biofilms increase the opportunity for gene transfer. The gene transfer converts a virulent commensal organism in the biofilm to a highly virulent pathogen. Quorum sensing, which we most of us already know, is a very important property of biofilms through which bacteria communicate. Bacterial species communicate with each other within the biofilm through quorum sensing. As the density of the biofilm, the cells increases, the organisms secrete low molecular weight molecules that signal when the population has reached a critical threshold. Quorum sensing is responsible for expression of virulence factors and helps in the survival of the pathogens. Now, biofilms may contribute for a development of altogether new virulent phenotype because organisms growing in the biofilms, they, uh, they have not been detected in the past. The organisms were when grown on rich nutrient media under planktonic conditions. So in the biofilm, the organism behaves altogether differently as compared to it is grown in a planktonic condition. Another property which makes biofilm very challenging is that the biofilms, they protect the organisms from immune responses, both immunological and non-specific defense mechanisms of the body. The contact with the solid surface triggers the expression of a panel of bacterial enzymes which catalyze the formation of sticky polysaccharides that promote colonization and protection, because biofilms, they are formed either on the living or the non-living surface. So the expression of these sticky polysaccharides, they protect these organisms from the immune responses. Now, coming to some of the important nosocomial infections which are directly related to biofilms, like for example, cystic fibrosis. In cystic fibrosis patients, they have Pseudomonas infection. Pseudomonas aeruginosa infection is very common that often results in antibiotic-resistant biofilms. Similarly, eye and biofilms. The presence of bacterial biofilms has been demonstrated on many medical devices or materials relevant to the eye, like contact lenses, suture material, intraocular lenses, and many ocular infections often occur when such prosthetic devices come in contact with or are implanted in the eye. Microorganisms may attach and develop biofilms on the components of mechanical heart valves and surrounding tissues of the heart, leading to a condition known as prostatic valve endocarditis, which is a very common complication in those having heart valves. The primary organisms responsible for this condition are Staphylococcus epidermidis, aureus, Streptococcus species, gram-negative bacilli, diphtheroids, and Enterococci and Candida. And at times it has been seen that these organisms may originate from the skin, other indwelling devices such as central venous catheters or even dental work, and enter the bloodstream and cause and form the biofilm on the mechanical heart valve, which the patient has got artificially transplanted. Now, urinary catheters and biofilms. This is again a very common problem in the patients in the hospitals, that urinary catheters, which are made of tubular latex or silicone devices, which when inserted may readily acquire biofilms on both the inner as well as outer surfaces. The commonly occurring organisms on them are Staphylococcus epidermidis, E. faecalis, E. coli, Proteus mirabilis, Pseudomonas aeruginosa is very common, Klebsiella pneumoniae. And also, the longer the urinary catheter remains in place, the greater the tendency of these organisms to develop biofilms and result in UTI. Now, the topic which I'll be focusing more is dental plaque as a biofilm, and what is its pathogenesis, and what are the diseases the recent research has established, and how to prevent those diseases. Now, dental plaque is a yellowish biofilm that is built upon the teeth. So, dental plaque is considered as a biofilm, microbial biofilm. It has a very much microbial etiology. And before I start with the development part of this plaque biofilm, what is the composition of the plaque biofilm? It constitutes both the organic component as well as inorganic component. The organic constituents of the plaque biofilm are polysaccharides, glycoproteins, lipids, and proteins. Polysaccharides, around 30% of the plaque biofilm, they are produced by bacteria, and their role is mainly in plaque retention. The glycoproteins, which are acquired from saliva, are important component of pellicle that coats the clean tooth surface. The lipids constitute 30% of the plaque and is derived from the membranes of bacterial cells, host cells, and food debris. Protein also constitute 30%, and albumin, which is derived from the saliva. Inorganic constituents of the plaque biofilm include calcium and phosphorus with trace amounts of sodium and potassium. These components are mainly, if it's a supragingival plaque, it is mainly saliva from which they are derived. The source of inorganic component of subgingival plaque is derived from the crevicular fluid, which is the serum transudate. Now, it's not that only bacteria constitute the composition in the plaque. There are mycoplasmas, yeast, protozoa, and viruses, fungi, and 1 gram of dental plaque contains around 2 into 10 raised to power 11 bacteria. If I come to the development of this plaque biofilm and briefly I describe it, then the first step is a reversible adsorption of bacteria on the enamel of the tooth, which is within seconds. The second step is the irreversible attachment of bacteria, takes from seconds to minutes. And the third step is the growth and division of bacteria, increasing the diversity of the bacteria and takes place in hours to days. And the next step is the EPS production and a proper biofilm formation, which is a matter of hours to days. And lastly, the mature biofilm takes around days or months to develop. So a proper biofilm, plaque biofilm, if I talk about, takes days or months to establish itself on the tooth enamel. The formation of dental biofilm, let's take it step by step. The first step is adsorption of host and bacterial molecules to the tooth surface to form a conditioning film. This is like the host here is the enamel, and it has the various receptors on it, and the bacterial molecules too have adhesions on them, and they combine together to form a conditioning film, which is called the acquired pellicle. So, like I've taken 1 or 2 examples. If I take the bacteria like mutant streptococci, it has the adhesion glucan binding protein, and the receptor which is located on the enamel is the glucan. The second step is, like, once the conditioning film is formed, the transport of microorganisms to the pellicle coated tooth surface. The third step is long-range physico-chemical interactions which take place between the pellicle coated tooth surface and microorganisms. That is the van der Waals force of attraction, they exist, and the adhesion between the microorganism and the receptor on the enamel is still at a reversible stage because of the weak forces of attraction. In the next step, there is short range of interactions between, or stereochemical interactions between the adhesion which is present on the bacteria on the microbial cell surface and acquired pellicle that is on the enamel. And now this interaction takes becomes irreversible. And also the immunoglobulin A proteases present in the saliva do aid in furthering this adhesion as irreversible. And simultaneously, oxygen is consumed by microorganisms and replaced with carbon dioxide. That is, aerobic organisms, they are using the oxygen and replacing it with carbon dioxide. And the co-aggregation in the next step, the co-aggregation, the diversity of microorganisms is increasing and resulting in the diversity of the plaque microflora. At the next step, that is, this is at this stage that the multiplication of the attached organisms produces a confluent growth and a biofilm which acquires a typical corn-cob appearance. And it is at this stage that the extrapolar polymeric substance is secreted by the bacteria and it acquires a biofilm appearance. And when this biofilm is almost mature or mature, the detachment of cells from the biofilm starts taking place into the planktonic phase, and these cells, organisms, they start colonizing the fresh sites. So this pic is showing the typical corn cob appearance seen on scanning electron micrograph. So what is happening? Metabolic changes in the biofilm is that the nutrients are being used up by the microorganisms. They are decreased. The oxygen concentration is decreased. The EH is from positive, it is becoming negative. The pH is going down. It is becoming acidic. The metabolic products are released by the bacteria. And the important thing to in a plaque biofilm is that this sodium, which ions, the concentration of sodiums, which was higher initially in a healthy mouth, it completely reverses and changes into more of potassium ions than sodium ions. That is, the environment from alkaline, it is becoming acidic. And if we see what is happening at the microbial level, so microbial level, if we see the early colonizers in the mouth, in the healthy mouth, they were mainly gram-positive cocci and they were aerobic in nature. And then when the new organisms, a biofilm starts establishing, the organisms become mix of facultative aerobes, that is gram-positive and gram-negative rods. And later on, the environment favors completely anaerobic organisms which are basically gram-negative rods. So we can see at the microorganism level, the flora is shifting from gram-positive to gram-negative, and this is what completely changes the oral microbiome of the mouth, or what is called as oral dysbiosis. So once again, I would like to stress upon what are the microorganisms as primary colonizers in the mouth. The initial colonizer species to dental pellicle are Streptococcus species, Haemophilus species, Neisseria species, which are obligate aerobes, and later on Actinomyces species, Veillonella species, which are facultative aerobes. So primary colonizers produce low oxygen tension that permit the survival and growth of obligate anaerobes. And then in the last stage, when the complete established biofilm is there, we can see altogether this microflora starting from aggregated bacter species, Campylobacter species, Fusobacterium, Prevotella, Porphyromonas gingivalis, and Treponema species are mainly obligate anaerobes in nature. So this is what, and cell signaling, like we already know, quorum sensing, it enhances the communication in different bacteria and further enhances the biofilm formation. So this was about the development of the plaque as a biofilm, the steps which were happening, which led to the development of the plaque biofilm on the tooth enamel. But what is the clinical significance of this plaque formation? See, there are two types of plaques which are formed. First is the supragingival plaque, which is when bacteria are adherent above the gingiva, that is at the top, the outermost portion, and it leads to diseases like caries and gingivitis. And the plaque which is formed in the tissues, that is bacteria adherent below gingiva, leading to periodontal diseases and other periodontal diseases. So dental caries is the disease known as tooth decay, consequence of dental plaque formation. When the plaque calcifies and leads to the dental caries, it is known as tooth decay or cavity, is a biofilm dependent disease, bacterial in origin, that causes demineralization and destruction of the hard tissues of the teeth. It could start from enamel, infect the dentin and the cementum. So we see that it is a result of the production of acid by bacterial fermentation of food debris accumulated on the tooth surface. Mainly, the organisms responsible for this are mutant streptococci and lactobacillus species. So these are the stages of dental caries. The plaque hardens to form a tartar, calcifies. The acid wears out surface of the enamel. This takes the time, as we can see. The erosion is faster in the dentin because it is comparatively softer, and erosion hits the pulp where nerves and blood vessels are. So it is at usually this stage, second or third stage, where the dentist, they can identify the cavity which is formed in the enamel of the tooth, and it is the first stage over here when a simple spot, a yellowish white chalky spot is formed that needs to be detected. And I think this is the area where scientists can work upon so that the filling of the tooths can be avoided, that how we can detect the caries at this stage, the initial stage itself, when there is a formation of yellowish white chalky spot on the enamel. That is just the beginning of the caries. Now, factors causing caries. There are four factors which are responsible for causing caries. First is the susceptible tooth. In this, you have the what is the saliva, because individually everybody has a different quality and quantity of saliva, and also this saliva can be affected upon various diseases a person is going through, like xerostomia may be the result of a head and neck cancer which a patient has. And also the structure of the enamel, we always see that some people are more susceptible to caries, and they commonly say that my tooth enamel structure is like that. So the susceptible tooth is a contributor to caries. The second is sugar, which is very important part, the amount of sucrose, because these bacteria adhere to the sucrose, and sucrose is an important factor in binding these bacteria. The third is the bacteria. Now, the presence of certain bacteria, for example, let's say mutant streptococci and lactobacilli species in the mouth, it in some people which have more numbers of these bacteria, obviously they are susceptible more to the caries. And the time is also very important factor in causing caries, because time is directly related to the frequency of the carbohydrate or sugar consumption. A person who has more frequency of consuming sugary substances is likely to get caries more as compared to a person who is taking sugars at a comparatively bigger interval. So the next is again disease, which is a consequence of plaque formation, which is the inflammation of the tissue, that is the gingivitis, a non-destructive type of periodontal disease, which left untreated can develop in periodontitis. And this is essentially caused by gram-negative bacteria. As we were seeing in the dental plaque biofilm formation, that gram-positive are changing into gram-negative bacteria. Compared with healthy sites, noticeable increase in cultivable and uncultivated cultivable spirochetes are also detected in this, and there is a dramatic increase in Prevotella intermedia in gingivitis. So these are the changes at the microbial level etiology which causes the gingivitis. Periodontitis results from untreated gingivitis. When gingivitis is not treated, it can lead to periodontitis, and eventually it is the tooth loss which happens. Gram-negative, and speaking from the microbial perspective, gram-negative bacteria are more pronounced in chronic periodontitis. The virulence properties are increased, and the etiological agents, mainly the bacteria which are found in this chronic periodontitis, are P. gingivalis, Tannerella forsythia, and Prevotella intermedia, and Fusobacterium nucleatum, which are obligate anaerobes. So these were some of the diseases which we already know are caused by dental plaque biofilm. Uh, but oral health, what I want to specify in today's presentation is that oral health is directly linked to general health, and it is not just limited to the oral cavity. Now, recent scientific research has established a link between mouth infections and serious medical problems. And people have, although pilot studies are there available, not much data is there, but pilot studies have been done which link oral health with improper oral health with diabetes, rheumatoid arthritis, stroke, oral cancer, heart disease, and Alzheimer. Even now, only in 2020, it has been proved that even oral health, a bad oral health, is also linked to the development of Alzheimer's disease, that is a form of dementia. And mostly these associations are related to inflammation, as inflammatory markers are increased because of bad oral hygiene, and consequently they give rise to the severe forms of these diseases. For example, if we take cardiovascular disease and oral health, gum disease harms the heart too. And there are two theories which are being proponent to this. One is that the Porphyromonas gingivalis bacterium can actually migrate from the mouth to the heart and cause inflammation in the arteries. That means it leads to chronic inflammation of the arteries, and that promotes the atherosclerosis. And another is that people living with the chronic infection, such as a gum disease, because what is periodontitis, that is again a chronic inflammation, may produce higher levels of C-reactive protein, which could place them at a higher risk of heart attack. And it has been found that higher number of periodontal pathogens like Tannerella denticola and C. rectus, P. micros, Tannerella forsythia have been found out, isolated from people having these endocarditis and different forms of cardiovascular disease. And in fact, the guidelines of European Society of Cardiology has officially included that periodontitis is a risk factor for developing the cardiovascular disease. Another important disease which has a link with periodontal disease or periodontitis is diabetes. Now, persons with diabetes are at a greater risk for periodontal diseases. In fact, it is bidirectional. Patients with diabetes have increased risk for dental caries, oral candidal infections, and xerostomia. As we already know that people who are having diabetes, they contract more of infections. So that leads to increased risk of dental caries, oral candidal infections, xerostomia. And it works the other way around also. Like if they are having more of infections, that is going to lead to further disruption in the more of the hyperglycemic stage, which the person is already having. So untreated periodontal disease in persons predisposed to diabetes make them more likely to develop the disease. And it has been shown that effective treatment in these patients, once they were effectively treated for periodontal infections, there was a reduction in periodontal inflammation, and it was directly related to a reduction in glucose blood levels. So periodontitis and the blood glucose levels in a diabetic person is directly linked, and that has been now well established. Another disease which is directly linked with oral health, bad oral health, I would say, that is cancer. Because human papilloma virus HPV, herpes simplex HSV, they play a role in oral cancer development. 2/3 of oral cancers which have been studied, they have HPV DNA in their cells. So DNA from Epstein-Barr virus, cytomegalovirus, herpes simplex, HPV have been detected in oral cancer biopsies. And the second type of infection which leads to oral cancer is chronic candidiasis. It has been implicated as a risk factor in oral cancer. And it has also been found that certain strains of Candida albicans produce nitrosamines, which are carcinogenic in nature. So infections of the mouth, which are a consequence of bad oral hygiene, are also directly linking it with oral cancer. Now, this is something which has very recently come up. In fact, this has been quoted. It has from article which happened to be published in July 2020 in Frontiers. And they have, in a study conducted, they have clearly established that there is a direct relationship between periodontitis, a chronic inflammatory disease, and Alzheimer's disease. Alzheimer's disease, as we know, is a prevalent form of dementia in elderly population, particularly globally, and in today's lifestyle, it is becoming more and more prevalent. So they have proved that existence of a direct link between neuronal and non-neuronal inflammation, infection, and Alzheimer's disease progression. And this has been shown by P. gingivalis, which is the main organism in in periodontitis. It breaches the bloodstream and enters the brain, and it has capability of producing specific amyloid plaques. And these amyloid plaques, they can trigger the dementia, or already they could, a person who is having dementia could contribute to more of its severity. Again, just like diabetes, this is a bidirectional form. Alzheimer's is contributing to bad oral hygiene is contributing to the severity of Alzheimer's, and Alzheimer's is getting developed due to periodontitis disease, or which is resulting from a bad oral hygiene of the mouth. So it's not that oral bacteria, all of them, they are causing disease. Only some of the oral bacteria, they are linked with health also. There are around 10 raised to power 11 bacteria per millimeter cube of dental plaque, and there are certain bacterial species which have been said, which are associated with good oral health, like Streptococcus sanguis, Veillonella parvula, and Capnocytophega crassus. And these are mainly non-motile in nature, unlike in the diseased form where we find more of the bacteria in motile forms. So after these diseases, I would like to stress upon the importance of a good oral hygiene. That is, prevention is always better than cure. Good oral hygiene habits will keep you away from most of the dental problems, saving you from toothaches and costly dental treatment. So by maintaining good oral hygiene, we can prevent the microbial diseases like caries, dental caries, gingivitis, periodontitis, halitosis, which is the bad breath of the mouth. And what are the basic steps for maintaining oral hygiene? Although this is a topic of the dentist, but I would like to cover up in this because some of the students, they may not be fully aware of how to maintain the oral hygiene, or they are careless about maintaining their oral hygiene. So the whole purpose of maintaining oral hygiene is reduction in the microorganism load in the mouth. So first is brushing your teeth at least twice a day or after every meal. And second is flossing the teeth regularly. Proper diet, it plays a very important role in maintaining oral hygiene, interdental cleaning, rinsing, and dental checkups. So brushing of teeth, if always use a soft bristled toothbrush, use anti-cavity fluoride toothpaste, which are effective in removing the plaque biofilms, and hold toothbrushes at a 45-degree angle at the gum line. Brush in a circular motion, sweeps plaque out of the gingival pocket, and the teeth should be brushed for at least minimum of 2 minutes at least twice a day. And another thing which I want to highlight over here, like the brushing, of course, it should be done in the morning and the night. And usually we presume that the dental plaque is formed in the night, but on the contrary, more of the dental plaque is formed in the daytime. So in the daytime, rather, one should be more particular in keeping the oral hygiene. Brushing gums and tongue along with your teeth. Do not brush too hard because it can cause gingival gum recession. Second is flossing. You can get the methods of flossing. You can ask the expert for how to floss, but the reasons for flossing are: flossing removes plaque from behind the teeth that brushing misses. So there are some of the debris of the plaque and the plaque which the brushing misses. So that is removed by the flossing. The flossing helps prevent periodontal disease by removing the plaque. The proper diet is another factor. Am I audible? Okay.
Yes, you are. Yes.
Okay, thank you, sir. Proper diet is another thing which one should be very well aware of. That is, particular the young generation and all, which they take foods which are rich in high sugar content should be avoided. Like I had explained, how the caries starts with sugar is one of the important factor in which caries develops, and carbonated drinks which are more acidic than non-carbonated drinks, more dangerous. Sticky foods, they should be avoided, or proper oral hygiene should be maintained if one consumes them. Avoid excessive intake of fruit juices, which can be very acidic, or they can be diluted with water, and then intake should be there. There are various, for interdental cleaning, various interdental brushes are available, like single tufted brushes, which have one large bristle for cleaning spaces between teeth, to carry medicated gel in pockets. There are interdental brushes for cleaning spaces between the teeth, which are available. Interdental wood sticks are commercially available, which are specially shaped toothpicks made from either wood or plastic. Super floss, which is a special type of floss designed to clean beneath fixed bridges, so that they do not, the biofilm doesn't develop there. Then rinsing is regular rinses with a good mouthwash is another very important practice to be followed, which helps to keep mouth clean, fresh, and germ free. Any good quality mouth rinse which having chlorhexidine can be taken. Daily rinses must be alcohol free because they cause dryness of oral mucosa. The fluoride rinses can be done in those persons because they help to boost strength of a newly erupted teeth. So regular dental checkup should be part of your follow-up, and visit the dentist regularly to prevent any problem before it becomes hazardous. Regular dental checkups are essential and part of dental hygiene for both young and old alike. So it's not like that if any person is young, he or she doesn't require the regular dental checkup. And the signs of a good oral hygiene is that the teeth are clean and free of debris, the gums are pink and do not bleed on brushing or flossing, and bad breath is not a constant problem, because bad breath or halitosis is again a sign of some oral infection or a oral disease emerging in the cavity, oral cavity. So take home message from this is like the formation of a dental plaque biofilm. The consequence in terms of various diseases is that you cannot be healthy without maintaining good oral health. So this is the what I wanted to deliver in the presentation. But lastly, I want to thank my teachers and a very belated Happy Teachers Day. And you can see Professor Chibber and Professor Praveen Rishi, Professor Vijay Prabha, Professor Geeta Shukla, Professor Prince, and Professor S.K. Soni, Professor Kusum Harjai in this pic. I want to thank all of you for being my teachers, and it's really been very encouraging and motivating. Always, like today also, I feel very encouraged and motivated by your all of your words. And lastly, I want to thank the organizers, Professor Prince Sharma, who is the president of AMIU, the Association of Microbiologists of India, coordinator Professor S.K. Soni, who is the vice president, and Dr. Naveen Gupta, the very dynamic coordinator and secretary of AMI, and Professor Kusum Harjai, HOD Department of Microbiology. Thank you very much. Thank you all. Yes.
Dr. Naveen, your mic is off. Dr. Naveen, your mic is off. Mic is off.
Yes. So thank you very much, Dr. Sonia, for
Thank you, sir.
very informative with beautiful slides.
Thank
I will
you.
now again invite Tibber sir to initiate the discussion on this and any questions if he has in mind and all that. Tibber sir?
Okay. Okay. Sonia, very well delivered lecture. Congratulate you first of all.
Thank you,
Um,
sir.
I have few queries regarding this. One is the bacteria which cause dental caries. As you have already said, there are many of them, and the shift is from aerobic to anaerobic. Are these endogenous bacteria, or these are the ones which are from outside?
So
Because
mainly they are endogenous bacteria. Like what is happening in this all oral dysbiosis is that the numbers. I think, am I audible?
yes, yes, you are.
Yes.
Yeah, it is the number of bacteria which is increasing, and that is increasing multiple times in thousands and teraspasic standards. So it is the number of bacteria which is increasing. These bacteria may also be present in our oral healthy mouth as well. And also some of the bacteria, like for example, you have taken the example of caries, like mutant streptococci or lactobacilli. Although they are present in the mouth, some of them can also be acquired through horizontal transmission, which is there in the already a person who's having caries, and within the family also the child may be prone to more of the caries disease in the siblings itself. But yes, they are mainly of endogenous origin.
And second thing you said is motile versus non-motile bacteria in causing the oral, the biofilm formation and things like this. Can you explain this to me? What is the basis of this?
So initially, what they have seen in the biofilm formation, that in a healthy mouth, mostly these bacteria are non-motile. But as a biofilm or a dental plaque is built up, the mostly bacteria which are found, maybe the altogether the oral microbial biome is changing, and that could be the reason that initially the because the ones which are non-motile are different. And now what we find, the outgrowth of the, it's not like poriformis gingivalis is not existing in the mouth, it is already existing in the mouth, but in the periodontitis condition the numbers increase in multifolds. So that could be the reason that when they define, it could be from a non-motile to motile bacteria,
Mm-hmm.
because the bacteria altogether is changing.
Mm-hmm. My last question is in our in India we usually use neem and datun and things like this. Are
Yes.
there any reports which have shown any effect of these herbal thing compounds on the biofilm formation?
So, like traditionally methods, neem everybody knows has got antibiotic, antiseptic properties, and people are working on this direction. In fact, a lot of literature is there, so which says that these herbal medicaments, they are effective in removing the biofilm. But the question is that every medicament, herbal medicament, whatever it is there, has a different efficiency level. So neem already our tradition from thousands or hundreds of years back. Everybody knows it has got the antimicrobial properties. So yes, they are effective, very much effective, I would say.
No, my point is this is a very good field. I mean, one should explore this because many a times we know that this is causing this effect, but
Yeah.
scientific evidences are not usually available.
Hmm.
So I
And
think
the
the people like you should work on such things where you should explore why it is happening and how this can be increased. The biofilm deterioration or biofilm, to remove the biofilm from the dental implants or the caries. So this
yeah,
is a very exciting field. I think one should look into this. So
yeah, it's a
any
very
other?
challenging field. I'll
Yes,
definitely
yes,
try. I'll definitely try.
yes, yes. Good. Any
Thank you, sir.
other question? Any other question? Yes,
So,
yes, Dr.
Yes.
Alia. Yes.
Yes, doctor.
so
I
Good
have
afternoon, Dr. Alia.
four questions.
Okay,
Sure.
sir.
yes,
Most of them may look trivial, but I would like that if you can throw some light on them.
So the trivial
Your
ones are more challenging. So the
your
trivial ones
your
are more challenging.
your area is amazingly painful. Actually, gives more pain than anything else.
Okay, yeah,
Initially, you told that there is a biofilm which are quorum sensing mediated.
right, sir.
So, are there other non-quorum sensing mediated biofilms also?
So if I talk about most of the biofilms, they are which are there in the nature, they have these corrosensing property as a very important property. But the biofilms like dental plaque biofilm, which I have studied extensively, that is quorum sensing in nature. So there could be some non-quorum sensing also, but to that point, I'm not much aware of that. But quorum sensing, since it's an important property of a biofilm formation, what I think is like most of the biofilms, they are having quorum sensing as an important property.
Actually, medically speaking, it is. They say that 65% of the infectious diseases are caused by quorum sensing mediated biofilms,
Right,
which means 35% are non-QS mediated.
right. So
So,
most of them lie in the quorum sensing region.
yeah, no, most people study those which are quorum sensing mediated.
Right, yeah.
Question number two: how biofilms formed on plants are different from those formed on human beings?
So, like biofilms, the surface which is formed on the plants, the substrate level is different as compared to that in the human beings. Like if I talk about the oral mucosa, so there the surface is the enamel. So that way, and the more or less the development pattern is the same in the plant biofilm as a human biofilm, but of course the composition of some organisms is different. That in plant it could be more of the fungus and other organisms as compared to that of humans.
No, no, but as you say, the biofilm process is the same. It means that starting substrate has to be the same in both plants and human beings.
Yes.
Right, yes, yes, the
So,
substrates has to be same.
yeah. So, do you have any idea about?
Because, about plant biofilms.
Yeah, yeah.
So I haven't gone in plant biofilm direction. My field
Yeah.
of work has always been medical microbiology.
Okay. Question number three. So, are there any anti-biofilm agents which you know about for these plaques, anti-plaques?
So biofilm agents like the ones,
Anti-biofilm.
yeah, anti-biofilm, the ones in the last which I have discussed in the prevention part, they are rather the fluoride rinses, the mouthwashes, they are all anti-biofilm. Plus there are alternative strategies also to reduce the biofilm. One of them is antibiotics, the phages. They are there to control the, which are
No, no,
being tried upon.
madam, since you
Yes,
started with QS mediated biofilm, so which means we have to look for anti-QSI, huh? QS.
right, sir. Yes.
Rather than fluorides and antibiotics, which are conventionally known, and you say that the biofilms are resistant to antibiotics. These antibiotics are not solution. So we have to go beyond antibiotics.
Yes, we have to keep on discovering also anti-QS agents which can disrupt these biofilms.
Okay.
I
Question
think
number fourth,
Sir,
one small
Right,
question.
sir. Fine, sir.
I can add something to that. That there are many toothpaste which are beyond going beyond fluoride, and many of them they have the triclosan. Triclosan is a very known antibiotic compound, and you will find that many of the these toothpaste they contain triclosan. So I will say that triclosan-like compounds they are very commonly used in toothpaste and other things, and which have the antibiofilm properties here. Actually.
Properties.
And to the
Yes,
first
yes.
question, sir, your first question was that all the biofilms are quorum sensing mediated, or there are non-quorum sensing mediated
Non-sir.
also. So, as far as my knowledge is concerned, we have like for example, we were looking for quorum sensing in thermophiles, extreme thermophiles, an area which has not been looked upon. And we have the answer which we have till now got is that there are microbes in which there are quorum sensing mechanisms occurring, which we still don't know about them. We know a few classes of quorum sensing mechanisms, but we do not know about the quorum sensing mechanisms which are still existing, and we have to learn a lot about them. In thermophiles, there are mechanisms very, very rare which are existing, but we were not knowing about them. So I would say that it's very hard to say that all those biofilms which are produced by non-quorum sensing producing bacteria are they really non-quorum sensing producers, or we don't know them about the quorum sensing mechanisms.
No, but they say that is
Yeah.
cyclic AMP
Because
which plays,
Yeah. Yes.
cyclic AMP plays a parallel role to quorum sensing.
Yes.
So maybe that
so another
mechanism.
thing is that So another thing is that in the oral microbiome, like many of the organisms are still not being discovered. They are still uncultivable. So we
Yes.
really don't know some of
You
the
don't know
mechanisms.
how much of the microbes in the mouth of microflora. You don't know about them. You can't cultivate them.
Right. Yes, yes. That's what
Question
I'm saying.
number question number 4.
Right, sir.
You said that initially it is gram positive bacteria which quench oxygen, and then gram negative bacteria which cause the disease. So in that case,
Right.
Staphylococcus aureus, which is a very well established bacteria, how does it form a biofilm if this is the community shift?
So the community shift is more towards obligate anaerobes. Like they are existing as I already said in numbers before also, but their number is low. But because of this whole dysbiosis which is taking place, it is the numbers which increase, and that is why we say that it is going from the gram positive towards gram negative, because the predominant now is the obligate anaerobes. So the it's not that they are not there; they are also there, but the
Yeah,
predominant
because
population has shifted.
we S. aureus is very well known to form biofilm.
Right. Yes.
Right?
Yes.
If this
Yes.
transition
Yes. It's
happens,
from the skin. Huh. Right. I agree, sir.
okay.
Yeah.
I
Thank
hope
you, sir.
that. Okay, welcome.
Yes, sir.
Dr. Sonia, we have a question from our student also. Shine Arora
Yeah.
is asking you, doesn't the lysozyme in our saliva act against the biofilm forming bacteria in our mouth? What is the role of saliva?
Yes. Salivary action is very much there. Saliva is important for the mechanical clearing of these organisms, and in a way, they help. They try to reduce the biofilm to a certain extent. So, but it is when this pH of the mouth or the acid producing bacteria they try to dominate, that obviously the bacterial are going to overpower, and the biofilm formation is taking place. But yes, obviously salivary action is there, which is a defensive mechanism and is removing the number of bacteria in the mouth. It is aiding in removing.
Okay. Okay.
Okay, sir. So we have some questions from our online viewer. More questions because people are following us online. Somebody has
Right,
sent
sir.
me. Somebody has sent me this question on my WhatsApp. What I flashed is that after childbirth, some normal microflora develops in the mouth of the child. How these microorganisms help child from pathogenic microorganisms?
Yeah,
This
this
is
is
the
a very important. This is a very important question, particularly in the development of caries. Like a lot of people are working in this direction, because it is said that the child has very much inclination to get caries from the mother, and that is why it is stressed in the prenatal care that most of the time that the mother should be treated from caries. And more of the studies, in the studies also they have shown that if the mother was treated with caries, then the child had a very reduced incidence of developing caries, because these organisms for caries like mutant streptococci, it has not only vertical transmission, but it has also passes through horizontal transmission. So yes, they have from mother to child, it has got lot of pathogenic potential to develop the disease.
Okay. So another question somebody is asking, that in today's scenario of this COVID and all that, where you think the oral hygiene stands? How we can oral
Uh,
hygiene can.
a very good question again. Like what I wanted to take up this topic. There were two reasons. One, I'm from the past couple of years I'm working in the dental institute, and another thing, I think most of the people are not aware of that the oral dysbiosis could lead to the systemic diseases also. So in COVID scenario, as you know, the diabetic population and people having other diseases, they are more getting fatal stages of the disease, COVID, coronavirus or COVID-19. So in that scenario, I would just say that they should keep their oral hygiene at the utmost level. Okay, that is certainly going to benefit them from one of the protections from getting the complications of COVID, because in a way it is going to help in certain diseases, in controlling certain diseases.
Okay. So another very interesting question is that we are using lots of toothpaste, mouthwashes these days. If we
Hmm.
go back to our earlier days when जब हम दातुन करते थे, तो what is the comparison somebody is saying, as a dentist you draw between the two, that is that still you say that equally good or better than all the toothpaste which we are using these days?
So I would say, of course, the traditional methods have their own scientific reasons to do, and people who are actually, there are a couple of people in the dental area who are working in on this field of comparing the herbal treatments versus the toothpaste. They would more be in a better position to answer this, that whether toothpaste are more effective or herbal. But yes, nobody can deny this thing that herbal treatments or herbal methods have always are years old, and they have their own antimicrobial properties and no side effects and no toxicity. And that is one of the important, you can say, remedies where antibiotics fail or some other treatments fail. So it is always the most of the time the herbal remedies, they come at the rescue.
Yeah, I think that's good, as Dr. Chibbar also earlier said, that there is a good area where we can do some study with respect
Right,
to
sir.
microbiota and all that.
Hmm.
So, if any other questions we have from the team who joined us, or otherwise, I will request Professor Chibbar to give the final comments and summarize the lecture, please.
Yeah, Dr. Sonia, you have delivered a very informative and a good lecture,
Thank you.
making it very clear that how the microbes they play a role in the dental caries. Because
Right,
a
sir.
lot of interest has been generated during the past few years in the biofilms, and especially
Hmm.
their importance in the oral hygiene and the oral diseases has been highlighted, and you have clearly explained their relationship with other diseases as well, which, for example, the Alzheimer's, which is very recent observations. And I think people will be benefited from your today's lecture, especially when you have explained us how to take care of our oral health. So I congratulate you once again for delivering this lecture, a very informative lecture. Thank you so much, Dr. Sonia.
Thank you. Thank you, sir. Thank you all. Thank you. Yes.
So, thank you, everybody. Dr. Soni had to deliver the vote of thanks. He had to leave. So, on his behalf, I'm just saying that I'm thankful to all who joined live with us and who listened to the lecture, first of all. Then I'm thankful to Dr. Sonia, Chipper Sara said, to give us a because I think she's the only dentist person who's related directly to the dentistry around us. So she was able to give us the first and fourth. Thank you, Dr. Sonia. I have special
Thank you,
thanks
sir.
for Dr. Timber, who has joined us today and conducted the today's lecture. So that was very useful for us. Then I'm again a special thanks to Dr. VC Kalia, who is showing lots of interest in our lecture series, and his presence is definitely valuable to us. And I'm thankful to the whole team of AMI Chandigarh unit, Professor Prince Sharma, our president, Professor S.K. Soni, our vice president, and the team from the various institutes for the continuous success of this lecture series, and especially the contribution made by our collaborators, Department of Microbiology, Chairperson Professor Kusumar Jai, and the whole team of the faculty member who are always ready to extend any help whenever it is required to conduct this series. So I'm thankful to all of you. I think we will meet again next Sunday, and I must tell you, all these lectures are available on our official Facebook page, as well as we are loading it on our official YouTube channel, where all of the people can follow this lecture after the live sessions also. So thank you everybody once again.
Thank you.
Thank you.
Thank you. Thank you. Thank you, sir.
Thank you. Haji, thank you, sir. Thank you for coming.